Today, I want us to revisit in a little more detail the RH blood group system. Yes, you heard that correctly, the RH blood group system. The RH blood group system is one of the most important blood group system that we need to know as a medical technologists, clinical laboratory scientists. We also need to know this as nurses, doctors, and even patients need to know and understand their blood group and the research section of it as well. And you may also be quite interested in knowing the details surrounding the RH antigen. So, students, regular people, physicians, people in the healthcare field, they need to have a good grasp of the RH blood group system. The ABO is important, and the RH is also important. If you know the ABO and the RH in depth, then you would have covered majority of the concepts that are needed for the foundation of understanding immunohematology, blood banking, transfusion medicine, etc. Now, let us get into a little overview of the RH antigen system. We want to look in brief on the biochemistry. But before we get to the biochemistry, let us see if we can do a quick recap as to what we already know about the RH blood group system. So the RH gene, they are non-glycolated protein. Basically, this means that no carbohydrates are attached to the protein. All right, that's the simplest way to explain it. The RH antigen reside on the transmembrane protein and are integral part of the RBC membrane. So if we know this and we understand this, then we are better positioned to appreciate their involvement in the structural integrity of the red blood cell membrane. The gene produce, well, let us say the gene products of the RHD and the RHCE are remarkably similar in that both of these two, I look at them as twins. You know, both of these encode for protein, which compose about 416 amino acids that trans that traverses the red blood cell membrane about maybe 12 times, right? So there are differences, yes, in their expression, but I want you to understand that their protein differs only with about 32 to about 35 amino acid. Amino acid protein 103 is important for determining C, that's big C, or little c. That expression, let us think about the position 103. When we think of position 226, we are thinking about big E or lowercase e or little e. Only small loops of RH proteins are exposed on the surface of the red blood cell, and this provides the uh, conformational requirement for many different serological testing to differentiate between the RH blood groups and RH blood type, because you know we can have RH positive, we can have RH negative, we can have cis trans variation, we can have weak expression, weak D, we can have null, we can have mod. There are many variations which we're going to get into in a little while. So the RHD and RHCE protein and also the RHAG are exclusively on the red blood cell. And remember, that we indicated that they aid in the integrity of the red blood cell membrane, as they are transmembrane, transmembrane, transmembrane. So they traverse the membrane. It is not surprising that they play a very important role in the structural integrity of the red blood cell. There is one hypothesis that has been in circulation that, based on their structure, it appears that they may also be transporters. And this was postulated by Westhoff and colleagues, and they were trying to show a way in which they played a role in the transportation of ammonia. An alternative hypothesis is that they may be CO2 transporters. So these are still in the developmental phase, the hypothesis phase. So they still need to be explored a little more so that we can have a definitive as to the actual state of matters. All right. So with that in mind, we have a general understanding in parts, but it gets a little bit more complicated. I'm going to put on the screen now, so for your visual pleasure, RHCE right here. And we're going to also have RHD right here. So you can see what is happening. So RHCE, RHD, and we're going to put right in the smack middle, right here, RHAG. Now we're going to move the RHAG to the farther side and move the RHD into the middle. So you can use the RHD as your base marker and CE and compare it with the RHAG. So if you do that, you can see what is similar and what is different. So just take a quick look at them and see if you can pick up what is similar and what is different. With that out of the way, let us think about the antigen characteristics. So as mentioned previously, Rh antigen are protein which are integral to the RBC membrane, passing through the RBC wall approximately 12 times. The antigens are found only on the RBC and are not soluble or expressed on other cells. The Rh antigens are well developed at birth. So at birth, you come prepackaged with your Rh antigen, just like how you have your A, B, O blood grouping antigens in the forward already developed. You also have your Rh already developed at birth. So for this reason, we have to be very mindful, very vigilant, and very cognizant of the implications that are involved with a mother and a child having a situation where there is hemolytic disease of the fetus and the newborn. That's HDFN, which it is currently called. But prior to now, it was called hemoblastosis fetalis. It's basically the same thing, it's just a different nomenclature. So that there has to be taken into account, and it is thanks to the Rh why we have to investigate even further in order to have a knowledge or an understanding as to these nuances that exist in the blood and the likelihood of a reaction which can lead in parts to a termination. So we have to be mindful of all of these caveats that exist. Another thing that I want us to understand is that the Rh antigens are also immunogenic, and exposure to foreign antigen through transfusion or pregnancy can cause an immune response with the production of the corresponding antibodies. So when we say they are immunogenic, we are basically just saying that they have the ability to elicit or evoke a reaction, and that reaction would be to create antibodies against that which it is. All right. So if an Rh negative individuals come in contact with a Rh positive individual during transfusion or through pregnancy, then there can be development of the Rh antigen. The Rh antigen comes into contact, there can be a reaction taking place, and when the antigens come, what we create are antibodies, because usually it's the cell that is transfused, it's the cells that become compromised, so the cells are going to fight back and produce the antibodies, which can be detected by various testing methodology. The five common Rh antigens are the D, C, E. Those are uppercase, but we also have lowercase c and e. And their specific corresponding antibodies account for majority of the problems due to Rh antibody. As we had discussed previously, that the D antigen does not have a allele, but the C, big C and little c are alleles, and big E and big E is allergenic to little e, all right? So the frequency of some of the Rh antigens can be tabulated. What I want to bring to your knowledge is that there are many different Rh antigens. We have D, C, E, uppercase. Then we have C, E, lowercase. Then we have CE slash F. Then we have big C, lowercase e. We have CW, we have G, we have V, we have VS. So there are quite a few Rh antigen that we have to be aware of. In some instances, we can say that the CW is rare, especially in the African American community. For Caucasian and African American, we have the D, which is usually the biggest problem causer, all right? But that is not by itself. We have the C and the E uppercase, which do also cause problems if they are found, and we have little c and little e. Those are the major ones that we need to be aware of. So big D, big C, big E, and we have lowercase c and lowercase e. So we have five major ones that we need to bear in mind. So if we can bear these five in mind, we will position ourselves to understand that if we have one hand, we have five fingers on that hand. So we can now put it in our mind. D, the big thumb, bam, this one is not allele. Then we have C, allele, E, allele, E, allergenic to E, lowercase e. So we have that idea in our mind, all right? So just have that concept so that you can use this information to navigate the waters of the Rh blood group system. Now, as it relates to strength, immunogenicity of the common Rh, and we made allusion that there are five. You have one hand up, and when you put out all the fingers, you have normally five. So that there is what we're going to be using to remember. You have the ABO, you have the RH. So we're going to use one hand for the ABO and one hand for the RH. And with the RH, we know we have positive and negative. But when it comes on to the antigens, the major ones, five, D, big D, big C, big E. Then we have lowercase c, lowercase e. Simple as that. Now, as it relates to immunogenicity of the common Rh antigen, D has the greatest, then we have little c or lowercase c, then we have big E, and then we have big C, and then finally we have lowercase e. With that said, that brings us to the idea that though someone is D, they can be D positive, D negative. But sometimes it is not black and white like that, positive or negative. Sometimes we have a situation where there is a weakened expression of the D. So we have some classification we consider to be the weak D, which is a variation of D antigen expression. So when Rh positive RBC samples are typed for the D antigen, they are expected to show strong positive reactivity with the anti-D reagent, because you know that clear liquid that we always use in the ABO RH blood grouping for the forward procedure, that there is a D antibodies that are used. All right. So we understand that when Rh positive red blood cells are typed for the D antigen, we want to know if the D antigen is there, they are expected to show a strong positive reactivity with the anti-D reagents, and that there is antibodies we're talking about. So let us just try to break that down a little bit more. A strong reaction is 4 plus or 3 plus. 2 plus is considered on the middle ground because you have 2 plus, then you have 1 plus, and then you have 0, which is no agglutination. So 2 plus is where we become a little bit suspicious of what is taking place. But 3 plus is good. 4 plus is even better. That is undeniably positive, all right? But when it comes to 2 plus, we have to investigate a little further, depend on the circumstance. However, some individuals have red blood cells that possesses variation in the quantity of the D antigen, because we know that the Rh, they are on the surface of the red blood cell and they are traversing the red blood cell. So they aid in structural integrity of the red blood cell. So if they are on the red blood cell, they are antigens. In circulation, we have antibodies. With that, we can go a little deeper into our investigation. So when the specificity of the D antigen epitope results in a weakened expression of the D when tested, by the serological method, that gives us an indication that there may be a weak D that is here, right? Beautiful. So serological weak D is noted when the initial D test is negative or less than or equal to 2 plus. So in some instances, the person can appear to be negative, but they are not true negative because they have the D, just that the amount that they have is so low that it is not reacting in a favorable way to give us a positive reaction. So that there is something that we have to be mindful of. So if it is negative, 1 plus, 2 plus, those are indication that there may be a weak expression taking place. So in order to deduce and detect and come to a conclusion of this state, we have to move on to indirect antiglobulin IAT phase of the testing. So advances in the RH testing methodology and reagents have resulted in detecting more weak D types without the need for the IAT. But there are still circumstances in which weak D testing is necessary. Individuals with red blood cell carrying weak D antigen can produce anti-D if they are missing the epitopes of the D antigen. And that is something we have to bear in mind. Now, for many years, the generic term of weak D was used to identify individuals who the D was not detected or was not detectable at the immediate spin without defining the nature of the weakened expression. Now, it is known that weak D can arise from several mechanisms. Yes, you heard that correctly. Weak D can arise from several mechanisms. Weak D types can be separated into three categories, and the three categories that we want to look at to make a mental image includes that of position effect, the quantity or the quantitative effect, or the partial D antigen. When we speak of partial D antigen, we're talking about missing one or more of the alleles. So those are the three main categories of the weak D separation. So if we are separating the weak Ds, we're going to think about position effect, the quantitative, and the partial D antigen. So it has been shown that up to 2% of individuals in the Europe ancestry possess some altered D antigens. So if you are European, there is a probability that you possess some altered D antigens. But that doesn't means that something is totally wrong with you. It's just that it's genetics. Based on where you're from, where you're derived from, there is a probability that you may be predisposed to a particular phenomenon. And for you, this may be such a phenomenon. It's only 2% of individuals. But if there are 2% of you, that's a lot of you, right? So when we think about altered D antigen, this may occur more often in individuals of the African descent. So you may have thought that I was only talking about Europeans in our session, but no, we are mentioning the Africans as well. So persons from the African culture, there is a big distinction when it comes on to blood banking, immunohematology, of the Africans and also the Europeans or the Caucasians. There are similarities with the prevalence of certain blood group system, but there are distinction when it comes on to other blood group system. So we have to be very mindful when we are classifying someone's blood group system, and we have to verify the appropriate race. So when your primary care physician asks you to indicate your race, as I indicated before, it is not necessarily racial profiling. Yes, there are some instances where racial profiling is done, but in most instances, it is not for that purposes. It is just so that they can give you and aid in determining the best course of action based on what is the norm or what is the consensus of expectancy for a particular race. So altered D antigen occur more often in individuals of the African descent, all right? But the exact prevalence is not quite known. And we have other situation which may complicate things a little bit of individuals who possess the D epitope in their RHCE protein. So that there gives us a little complexity in our navigation of this type of phenomenon. There is something that I want to bring your attention to, and that is the position effect. We made mention of it briefly, but we're going to make mention of it a little bit more in depth so that you can have a grasp as to what it entails. So the first mechanism that many have postulated and indicated that may result in a weakened expression of the D antigen was originally described as a position effect or the gene interaction effect. Basically, what's happening is that the allele carrying the RHD is trans. When we say trans, we're thinking about opposite haplotype, right? They're in the opposite haplotype to the allele that is carrying the C. So let us put that into perspective. Let us take the positional effect of C in trans to D. Let us use an example. Let's say we have DC, capital D, lowercase c, lowercase e, and then we have lowercase d, capital C, and lowercase e. The Rh antigen on the red blood cell is normal, but the steric arrangement of the C antigen in relation to the D antigen appears to interfere with the expression of the D antigen. So this interference with the D expression does not occur when the C gene is inherited in the cis position to RHD, such as D, capital C, lowercase e, slash lowercase d, lowercase c, lowercase e. It is not possible to serologically distinguish genetics or genetic weak D from the position effect weak D. So in order for us to do this type of deciphering, the immunohematology department and the molecular diagnostic department has to work in tandem with each other to come to the realization and the conclusion as to what is occurring and the best course of action to take based on the fact that the serological test can take you to point A, but in order to get to point B, you need a molecular testing. And to go to point C, if needed, then we will need a little bit more advanced testing methodologies. So practically speaking, this is unnecessary because the antigen is structurally complete. So if there is a situation like this, we don't necessarily have to do molecular testing, but if we need to find out for research purposes so as we can write those PhD thesis papers, then this would be something to explore. These individuals can receive the positive red blood cell without any adverse reaction. So yes, there is a positional effect where we don't get the result that we actually expect, but there's nothing really wrong with the person. The person is still classified as D positive. Now, We have the weak D and we have the quantitative changes due to fewer D antigen sites. We have spoke in brief about the pro-zone and post-zone reaction when you have too much antigen, too much antibodies. In the real sitting of an individual whose body produces only a small amount of the D, then the quantity that is present is not sufficient to give the reaction expected using the commercially available reagent. So the second mechanism results from the inheritance of RHD gene that codes for the weakened expression of the D antigen. So it doesn't mean that it's not there. It just means that it is weakened in its expression. So you are still there, but because you are so small, nobody is seeing you. So you have to grow a little bit bigger so that people can see you, or you have to multiply your territory so people can acknowledge your existence. It's the same thing in the body, how the body functions. So the D antigen expression, or that is expressed, appear to be complete, but guess what? Fewer in number. So they have everything that they need to have, but they're just fewer in number. You have persons who are of normal height, you have persons who are of the dwarfism, and you have persons who are of gigantism. So it's just basically the quantity. That's basically it. It's nothing complicated. It's simple. Just bear these concepts in mind so that you can navigate understanding the RH blood group system. On a molecular level, mutation in the RHD gene occur in some instances, and this causes change or deletion in the amino acid present in the transmembrane or the intracellular region of the RHD protein, thus causing conformational changes in the protein. So with that, we have an understanding of what is basically happening. So when we think about this, we want to think about individuals with weak D phenotype, rarely express or rarely makes the anti-D, since the RHD antigens are present as expected, but the protein changes occur inside the red blood cell. On a molecular level, changes in the RHD genes occur, which causes the amino acid present in the transmembrane region of the RHD protein. All right. So mutation in the RHD gene can cause this altered expression, and then we can classify it in category 1 and 3, which is usually the ones that are most common. You have other categories, but we're not going to get into that just yet. The next thing that I want to bring your attention to is DL. So DL is a phenotype which occurs in individuals whose red blood cell possesses an extremely low number of D antigen sites that most reagent anti-D are unable to detect. So in order to detect these, we have to do some absorption and elution of the D from the individual's red blood cell in order to detect the D, right? The presence of these can be determined using a 37-degree methodology. The 37-degree incubation provides time for the anti-D to bind the few D antigen sites present on these red blood cells. So molecular studies can be used to determine or detect mutation. This phenomena is allegedly rare in Caucasian. DL individuals are usually from Southern East Asia descent. Yes, Southern East Asia descent. have not been reported actually to make anti-D in response to transfusion or pregnancy. But there has been fewer instances of D antigen production in Caucasian individuals. Right now I'm going to put up on screen two three-dimensional illustration of weak D and partial D. And this here is taken from the Technical Manual 17th edition, AABB, and this is page 389 to about 410. I'm just going to put these two models on the screen, and then you can look what is similar and what is different. So here we have the weak D, we have the partial D, and we have the external or the exterior, we have the plasma membrane in the middle, then we have the interior, then we have the type 1, type 2 in the D, and you can just look at it and see if you see anything that is similar and anything that is different. There is something else that we have been privileged to understand, and that is the partial D or the D mosaic. This is a third mechanism in which the D antigen expression can be weakened. And this one is when one or more of the epitone within the entire D protein is either missing or altered. So this is termed partial D. And sometimes we call it D mosaic. Just try to be a little bit fancy. So the D antigen is not complete because one or more epitopes are missing. So if it is not complete, guess what's going to happen? It may evoke a reaction to the parts which are missing. That's correct. So the D antigen is not complete as we stated. So just bear that in mind. Partial D, not complete, because one or more of the epitones are missing. The serological detection of the D antigen varies greatly for partial D individual. Some individuals, you see, have red blood cell within the partial D antigen that may type weaker than expected, or they may not react at all when the routine procedures are used with most common commercial anti-D reagent. Other individuals have partial D and they are type normally with reagent anti-D. All right. The partial D antigen can be classified on a molecular level and are attributed to a hybrid gene resulting from portion of the RhD gene being replaced by a portion of the RhCe gene. The resulting protein contains a portion of the RhD and RhCe in various combinations, deepening the classification of hybrid gene makeup. So the anti-D made by individuals expressing the partial D can cause hemolytic disease of the fetus and newborn. And you understand why this can happen, because it is not complete. So some parts are missing, and because some parts are missing, giving the positive blood to the partial person can create a immunogenic reaction to the parts which are missing. So this can cause also HDFN and transfusion reaction, or even both. So once anti-D is identified, Rh negative blood should be used for transfusion. So we need to note that the anti-D produced is an alloantibody because it is directed against an antigenic epitope that the patient lacks. So it is not the patient who is just getting up and producing it. All right, so we have to understand that. So with that said, we are going to pause this section, and in the next sitting, we'll go a little bit deeper into the D epitope on the RHC protein.